Hi, my name is Thomas White.
My name is Campillo, and this is Dr. Ramos' lab. Dr. Ramos' lab is looking for the line 1 protein in lung cancer cells. Line 1 is a jumping gene. It can accounts for 17% of the whole genome. Line 1 is very mutagenic and contributes to the development of several cancers. Okay, the lab is focused on blocking the line 1 protein. The protein is believed to be active when an individual is in the womb. When a person is older, the protein is not as active as it was when the individual was a newborn. However, when a person smokes, this protein is active and affected much more than the proteins that are present within the lungs. The question that the lab is focusing on is how is it affected and why.
In order to answer these questions, the lab must first be able to identify the line 1 protein. The tool that helps us identify the protein is a citation imaging reader. The citation is used to read the concentration of mice tails, proteins, and to help culture cells. The questions that we have about the citation machine is why do we need the concentration of the DNA and the protein, and what does the concentration tell us? The first step is to calibrate the citation machine with water. This is done by injecting water into the well plates and then having the machine read it. Next, the wells are loaded with 2 microliters of DNA from the mice's tail. After the DNA has been loaded, it is placed into the citation and the sample is run. The citation gives us the concentration in nanograms. The concentration is needed in order to make the concentrations equal so they can be run through a gel and viewed under the UV light. After the concentration of the DNA is adjusted, 18 microliters of each sample is ejected into the agarose gel. Under the UV light, the protein is much more visible and shows us which mice have the protein we are looking for.
Okay, so the ending questions that we have are, would the concentration of the DNA and other proteins be able to make a medicine stronger than it already is? How is the medicine produced after the concentration is known?